Alright, so let's continue on with the second video of chapter 14. The last thing we talked about was the fermentation reactions in order to regenerate the NADs. We're going to do an oxidation of pyruvate either in one step to lactate, and that's lactic acid fermentation in U and some bacteria, or in two steps to acetaldehyde and then to ethanol. That's in yeast. And so with NADH acting as an oxidizing reagent or reducing reagent, it becomes oxidized back to NAD. And so we can do another round of glycolysis. So now that we've talked a little bit about NAD, let's go ahead and talk about NAD and why we can do these high energy electrons in NAD. And it has everything to do with the structure of the nicotinamide allows for a redistribution of electrons. So when NAD is reduced to NADH, it can take on these two electrons and redistribute them across many bonds in the nicotinamide. That allows for some high energy electrons to be carried from the reactions, whether they are in glycolysis or during TCA. Take these high energy electrons from those reactions and shuttle them to the electron transport chain where we can get the energy out of them. So it has everything to do with the redistribution of electrons across the nicotinamide. Okay, so after we've generated these NADHs or FADH2 if it's from the citric acid cycle, then those electron carriers are going to enter into the electron transport chain. So what is the electron transport chain? It's going to take these electrons because complex 1 NADH dehydrogenase has more affinity for those electrons than the NAD does. It's going to take those two electrons and use that energy of those electrons to pump a pair of hydrogens from the matrix into the intermembrane space. And then from complex 1 to ubiquinone to complex 3 to complex 4, each step we're going to have the electrons falling down in energy, and that energy being used to pump hydrogens across, whether it's 2 electrons in complex 1, 2 hydrogens in complex 1, or 2 hydrogens in cytochrome BC1 complex, or 1 hydrogen in the cytochrome oxidase complex. Each time we're going to pump hydrogens by the electrons falling down in their energy. And each complex has a little bit more affinity for the electrons, so that the shuttle from one complex to the next is energetically favorable, because each complex has more affinity for the electrons. So the electrons are falling down in energy level by moving from the least affinity to the most affinity, and then that energy is going to be used to move hydrogens across the membrane to create a hydrogen gradient. This is all going on in the membrane of the mitochondria. So mitochondria, there's a endosymbiotic theory that these were rickettsia, that some bacteria or some cell that was 2 billion years ago was not able to participate in aerobic reactions. So an anaerobic eukaryote. And it swallowed this rickettsia, but for some reason didn't digest it. So now that that bacteria is inside of a phagosome. And that explains a lot about its current structure. Mitochondria has a double membrane, an inner membrane and an outer membrane. The outer membrane probably being left over from its phagosome origins, and the intermembrane being left over, or the inner membrane being left over from its bacterial origins. The reactions are going to take place in the inner membrane space, so we call that the matrix. So the citric acid cycle is occurring inside of the matrix, and the electron transport is happening across the inner membrane, pumping hydrogens from the matrix to the intermembrane space in between those two membranes. So, so I'm not going to go through the steps of the Krebs cycle. I just want to point out that we're going to, for each turn of the TCA, we are going to produce 3 NADHs. We are going to produce 2 CO2s. We are going to produce 1 FADH2, and we are going to produce 1 GTP. So this whole process takes place in 8 steps, and the last step is going to produce deoxaloacetate that the acetyl-CoA binds to form the next citrate. So so it's a cycle. The input into this cycle is going to be acetyl-CoA. That acetyl-CoA could be coming from the oxidation of pyruvate from glycolysis, or it could be from fatty acid metabolism or protein metabolism. So all roads lead through vinegar. The acetyl-CoA is where all of these inputs are going to come in and enter into the TCA. So we are going to, like I said before, in this whole process, generate NADHs. Those NADHs are going to shuttle electrons to the electron transport chain, creating a proton gradient from the inner membrane space into the matrix. And that proton gradient is going to be the energy that is driving this ATP synthase. The energy is causing a rotational energy. So the energy of the proton gradient is going to create a rotational energy in the ATP synthase, and that rotational energy is going to cause conformational changes in 3 different subunits of the F1 ATPase. That's the lollipop head in the matrix side of the ATP synthase. And so at any given time, one of them will have ADP and inorganic phosphate bound to it, and then another one is going to be undergoing the reaction of turning that ADP and inorganic phosphate to ATP, which is an anabolic reaction, requires the input of energy. And then the third one is going to be ejecting the ATP into the inner membranes. So you want to look at the steps of the electron transport chain and look at the mechanics of how ATP synthase work. So both of those videos are on my website and also available in the web. So I've got 2 videos of each, one on the website and one in the PowerPoint. I suggest you go ahead and watch both videos and just become really familiar, really comfortable with how the citric acid cycle works, how the electron transport chain works, so that you can understand how molecules from food can lead to the acetyl-CoA entering into the citric acid cycle and generating NADHs that drive the electron transport chain, that create the proton gradient that drives the ATP synthase. So I would like for you to be really familiar with some bookkeeping, so that you understand how much energy is coming out of glycolysis, how much energy is coming out of the oxidative phosphorylation, where the NADHs are coming from. Really be familiar with the bookkeeping of how we end up with a lot of ATP. Okay, so that's going to wrap up chapter 14. So thanks for listening.